Hi, I'm Peter Jones, Associate Professor of Pathology and Lab Medicine at the University of Pennsylvania. Overall, the primary goal of my research is to determine how the extracellular matrix, or ECM, a cell-derived woven and globular protein network that envelops or contacts most cells within the body, an architectile of sorts, changes throughout development and disease, and how alterations in this extracellular matrix environment feed back to control cell and tissue behavior in 4D at the level of the genome and beyond. Towards these goals, and in the context of my collaboration with Jenny Sabin at PennDesign, we began by studying the influence of different ECM components, including laminin and type 1 collagen, on a number of nonlinear biological processes, including tumor formation, cell motility, and networking behavior in the lung. Ultimately, at the pathophysiological level, a major aim of our collaborative research is to derive new structural and functional information from each of these dynamic systems in an effort to diagnose, prognosticate, and ultimately treat human disease.
Hi, my name is Jenny Sabin, and I am a lecturer within the Graduate Department of Architecture at PennDesign at the University of Pennsylvania. My research and practice focuses on investigating the intersections between architecture, textile structures, computation, and cell biology. More specifically, this interest probes the hybridization of algorithmic design techniques generated through the analysis of biological design problems with experiments in fabrication and material construction. Through our collaboration, we aim to gain a rigorous understanding of how context or environment specifies form, function, and structure. The fabric world that this biological research interrogates unfolds examples of textile-dependent acts of nonlinear fabrication at the surface and at deeper structural levels. In parallel, this work offers up novel possibilities that question and redefine a contemporary notion of the architectile within the greater scope of generative design and fabrication.
Over the past 3 decades, it has been firmly established that 3D cell and tissue architecture, at the level of the extracellular matrix, exerts a dominant influence over gene expression. And in this sense, the ECM represents a key phenotypic determinant of cells within tissues. In essence, much of the secret of life resides outside the cell, within the extracellular matrix. And so to introduce the matrix, here you can see both in vitro and in vivo examples of cells derived from and residing within the blood vessel wall. In each case, these cells are surrounded by a globular and fibrillar network of proteins that are not only produced by these cells or their neighbors, but that also interact with these cells to inform their behavior. Importantly, the ECM not only acts as a physical entity, which lends flexibility and physical support to cells within tissues, but it also behaves as an informational entity, an ongoing and historical document that records what transpires in and around a cell during the lifetime of an organism. As can be seen from this slide, the ECM also forms part of an integrated network that extends from the cell exterior to the nucleus and back again via the cytoskeleton, and so it is relatively simple to imagine how structural and biochemical changes in the ECM might affect the deeper interior structure of cells. In a normal tissue, this type of cellular ecology is relatively static, but during development and with disease, the nature of the ECM and its interactions with cells changes radically. And when 3D tissue form changes, cell and tissue function responds with its own change.
Clearly, these biological models are underscored at the cellular and molecular levels by complex, nonlinear responses to complex and dynamic scenarios. Thus, we posit that a rigorous understanding and analysis of these types of models will allow architects to retool and reevaluate how we may negotiate topics such as nonlinear fabrication, feedback, and performance in architecture. On the architectural side, rather than seeking direct translation of the science to architecture and vice versa, this research collaboration is about working through biological design problems that give rise to new modes of thinking and working in design and biomedicine.
Towards these goals, our immediate collaborative plan was to expose architects to formerly unseen multidimensional ECM and cell-based biological systems generated in my lab at the IME, with the hope that this interaction would lead to both an extraction and abstraction of new biological information.
On the lab studio side, our initial aim was to introduce biologists to digital and algorithmic architectural tools that could be used to reveal new complexities within the biological systems being studied. Additionally, by specifically examining dynamic cellular systems, our intent was to discover new ways of understanding, revealing, and abstracting how these biological systems negotiate issues of auto and artificial fabrication. Here, structure and material are inextricably linked. As already mentioned, cells and tissues not only produce their own underlying, self-produced fabric, but they modify and respond to this woven, felted, or embellished environment. A digital code begets a physical code that informs the physical code, and so on.
Lab Studio, which we established at Penn in 2007, is one result of our ongoing collaboration. Within Lab Studio, 6 researchers, including postdoctoral fellows and graduate students at the IME, and graduate students in architecture and landscape design, were paired with one another. In the first phase, we chose to investigate surface design, motility, and networking behavior based upon new research investigating breast cancer and lung biology.
Initially, these co-investigations took place at the laboratory benchside, before being transferred back to the design studio. The advantage of formulating these scientist-architect pairings was that experiments could be redesigned based upon the architect's objective observations, intuition, and requests, and new tools could be developed and modified based upon the scientist's specific hypotheses. By working closely with one another, each scientist or architect could rapidly respond to one another's questions and needs in a relatively unimpeded fashion.